Welcome everybody to our webinar, Deconvoluting Compound Target Interactions with Limited Proteolysis. My name is Oliver Rinner, I'm CEO of Biognosys, and with me today are Paola Picotti, Professor at the ETH Zurich, and Nigel Beaton, Senior Scientist at Biognosys. So our mission, Biognosys' mission, is to provide cutting-edge proteomics technology to researchers so that they can solve their research questions and, for example, can move their drug development programs forward. We invent and we develop novel technology, especially in the field of data-independent acquisition for deep and reproducible protein quantification. And many of these developments that we provide as a contract research service or as software and reagent products arise originally from collaborations with leading scientists in academia. And LiP is such a collaboration example where we go from breakthrough science to a method that we can now offer to scientists that want to identify the targets of their compounds. So the reason why we found the LiP technology so interesting and why we exclusively licensed it from ETH Zurich was that it promised to address a really hard problem with an ingenious approach to directly enable compound target deconvolution in complex mammalian cell systems. So that was the promise, and in the second part, we will hear from Nigel where we stand with this ambition. The other reason was certainly that it acts scientifically an exciting structural dimension to proteomics and provides a unique tool to gain new insights into biological mechanisms. I think this is a fascinating approach, and I'm looking forward to the presentation from Paola. And now I would like to hand over to our presenters, starting with Paola.
Thank you very much, Oliver. Thank you for inviting me to this webinar today. So I will start my talk with an overview of the steps that led us to the development of LiP-MS, the technology this webinar is about, and then I will present some applications. So in the field of proteomics, we are often interested in protein networks. These are systems of interacting components like the one you see here, where the components are proteins. And these systems can be as small as the one you see here. This is a signaling cascade, or they can be as large as a whole cell. And typically, if we are interested in understanding how these systems operate and how they are controlled, we apply perturbations. And ideally, we would like to see how the components of these networks respond to the perturbation. Now, at the protein level, there could be several types of responses to stimuli. The first type of response is a change in the abundances of the components of the network. The second type of response is post-translational modifications, for example, protein phosphorylation. Then proteins can also change their assembly status, and they can change their interactomes, or they can change their subcellular localization. Now, if you look into the literature today, you will see that using proteomics, we can fairly well capture these four types of events at the protein level on a large scale. We can certainly capture abundance changes for a few thousand proteins in a sample using mass spectrometry. We can also study at least some post-translational modifications. And we can also study protein-protein interactions in a targeted manner using affinity purification-based approaches, but also on a global scale using size exclusion chromatography techniques. And finally, we can also look at changes in the subcellular localization of proteins using proximity tagging techniques. So I would say we are doing pretty well. We can capture these four types of interactions directly, of responses directly from a complex cellular matrix. But there is, in fact, a fifth type of event at the protein level, which is way less characterized. And this is protein structural changes. In biology, there are really beautiful examples of proteins that undergo a minor conformational change, and this is sufficient to trigger a whole cascade of downstream events. For example, think about a membrane receptor. When a ligand binds, this binding event is often sufficient to trigger a conformational change in the receptor, and this can activate a signaling cascade. Or think about an enzyme. When it binds an allosteric regulator, the binding event can change the conformational state of the enzyme, and this can profoundly affect the activity of the enzyme. So protein structural changes can largely affect protein function. And the question I would like to ask today with this webinar is, are we able today, or will we ever be able to measure also these types of events on a global scale, exactly like we do for the other 4 types I mentioned on the previous slide? But before I try to answer this question, I would like to tell you how we got interested in that. It all started back in 2013 when my lab was studying the molecular basis of protein aggregation diseases. These are pathologies involving the formation of protein aggregates, like the one you see here, in cells and tissues. And the current model is quite simple. One of these aggregation-prone proteins undergoes a structural change that makes it prone to stick to molecules of the same type. It forms oligomeric structures, and these structures can further mature and form larger aggregates, and these are also called amyloid aggregates. So we realized that when we started working on this topic that to study this problem, we really needed a technique to probe the pathological conformational transitions of our proteins of interest directly in a very complex biological matrix. We were at that time experts in mass spectrometry, and so we asked, can we use a bottom-up proteomic approach to study these conformational changes? So we reasoned that to be able to use bottom-up proteomics, we would need to first generate peptides that change when the structure of the protein changes. So some sort of structure-specific peptides. And then we thought if we manage to do that, then we can use mass spectrometry to monitor these peptides directly in a complex biological matrix. So we revived a relatively old technique. This is called limited proteolysis or native proteolysis. And it has been used a lot in the past, but mostly to study purified proteins or protein complexes reconstructed in vitro in the test tube. And our idea was, if we now couple it to an advanced mass spectrometric workflow, maybe we can apply limited proteolysis to a whole proteome. So now let's go step by step. I'll explain the technique. So first, in a limited proteolysis experiment, you use a broad specificity protease. For example, we like very much proteinase K, and you apply it to a protein substrate for a very short time, for example, 1 minute, and then you quickly quench the reaction, trying to capture the first cleavage sites. Now, the theory says that if you manage to do that, these sites should be located at the most flexible and accessible regions in a protein, because this makes the cleavage thermodynamically favorable. So this technique can, in principle, generate structure-specific fragments, because if the structure changes, the cleavage sites should be different. Then the main challenge was, how can we now apply LiP to a whole proteome or to a complex proteome sample extracted from cells and tissues? So we came up with a workflow you see here, which we call LiPMS. We first extract proteins from cells and tissues under non-denaturing conditions. That means we preserve protein structures. Then we apply proteinase K for a very short time, say 1 minute. At this step, we generate very large fragments, and these fragments are structure specific. They are, however, too large for bottom-up proteomics, so what we do next is to denature these fragments and then digest them again with trypsin. So from here on, it's pretty much the classical proteomic workflow some of you may be familiar with. In parallel, we also take an aliquot of the same sample and we digest it only with the trypsin under denaturing conditions as a control. So what is the idea here? The idea is that if you have a single protein and you digest it only with trypsin, you will generate an equimolar mixture of fully tryptic peptides. Now, if you add, before the trypsinization step, a limited proteolysis step, you will see that some of the fully tryptic peptides will be gone or show a decrease in intensity. And these are the peptides embedding an additional cleavage site by proteinase K. At the same time, you will see new peptides appear, and these are the 2 halves of the missing peptide in this case. So the main point about this technique is that it can generate proteolytic fingerprints, and these fingerprints are structure specific, and they can be measured directly from a complex cell or tissue extract for every detectable protein in the extract. So when we started first, we tested our idea on a protein called alpha-synuclein. Alpha-synuclein is a protein involved in Parkinson's disease, and it forms amyloid aggregates. You see them here when disease develops. So what we did was to prepare alpha-synuclein in two structural states. The healthy version of the protein, the monomeric form, this is a natively unfolded protein. And then we let this species polymerize into amyloid fibrils, and that's the pathological form of the protein, and these are rich in beta sheets. And then we spiked either this species or that species into a whole cell extract. This was a yeast cell extract. And then we asked, can we now detect this very small difference using our approach? So basically we prepared two samples that were identical. The only difference was the structural state of one single protein out of, let's say, about 4,000 or a bit more expressed proteins in this sample. So we applied the workflow I've shown you before, and this is the picture we obtained. So at that time, we saw that these two peptides, the blue peptides, were only detectable when we had the amyloid version of the protein in the lysate, while the green peptides were only detectable when we had the monomeric version of the protein. So what are these peptides? The blue ones map to the amyloid core of alpha-synuclein. So when the aggregates are formed, proteinase K cannot cleave anywhere here in this region. And so these peptides are generated only after denaturation and trypsin digestion. So they are canonical tryptic peptides. Now, when you have the monomeric version of the protein in the lysate, proteinase K cleaves here and here. The blue peptides are gone, and the green peptides appear. And these are the two halves of the missing peptides, from here on and from here on. So when we saw this picture, we got quite excited because this picture tells that we can detect two different structural states of a protein directly from a very complex biological matrix. Next, we wondered, well, does this approach work only for a very pronounced structural change? You can imagine that the structural change of alpha-synuclein is very large because it goes from an unfolded state to a very compact aggregated state. So we asked if it works also for more subtle structural alterations. Then we focused on a second model system. This is a protein called myoglobin, and it's a very nice system because you can prepare myoglobin again in two structural states, the holo and the apoform. And you do that simply by removing the heme group, the group you see here. Oh, maybe I have to indicate it here. And what happens when you remove the heme group? The helix F of the protein unfolds to a loop. So again, we prepared myoglobin in two structural states, and we spiked them into a yeast cell extract. And we applied again the same workflow. And like before, we saw that this peptide here was only detectable when we had the holo form of the protein in the extract, and these two peptides were only detectable when we had the apoform in the extract. Again, this peptide maps to the yellow helix you see here. It's the red peptide highlighted on the sequence, and it's present when the holo form is present in the extract because proteinase K has a hard time cleaving this region. And so this is again detected as a canonical fully tryptic peptide. When the helix is unfolded to a loop, proteinase K cleaves in the middle of the loop, and the two fragments of the larger peptide are detected. Now, if you look at the numbers on top of the sequence, these are the ratios between the intensity of a peptide in condition 1 divided by the intensity of the same peptide in condition 2. And as you can see, they are mostly close to 1 or maximum 2.3 everywhere except in this region where they are either very large or very small. And that's exactly the region that undergoes the largest conformational change, the region corresponding to helix F. So from this experiment, we learned that this approach can be used to locate the region in a protein that undergoes the structural change. Having gone so far, we also asked, can we use it beyond targeted application? So can we use it to identify proteins that undergo a structural change when we apply a perturbation to our system? So basically to address the question I posed on the first slides. And to address that, we performed a very simple experiment. We grew yeast cells either in glucose or in ethanol. That's a very well-studied metabolic shift. We then extracted the proteomes under native conditions, preserving protein structures. We applied the same workflow, and then this was followed by a data-independent analysis. So basically, we tried to measure proteolytic patterns for as many proteins as possible. We measured about 2,000 proteins in this experiment, of which about 300 changed the proteolytic pattern. And these were enriched in carbon metabolism enzymes. So we zoomed into carbon metabolism. That's the network you see here. And here you see in blue proteins that change their proteolytic patterns. And in red, you see proteins that increase their abundances when you switch yeast from glucose to ethanol. Now, even if you're not familiar with this network, something will be pretty obvious to you. All the blue proteins are on top and all the red proteins are at the bottom. So there seems to be some kind of modular regulation. And this is very interesting because these two groups of reactions have a peculiarity. The lower reactions, they just need to be a bit more active in ethanol than in glucose. And so this is achieved by increasing the concentration of the corresponding enzymes. While the upper reactions, they all need to be completely switched off or reversed when you switch yeast to ethanol. So it seems like the cell is using two types of regulation to achieve the different aims. We also picked some of these blue proteins and followed them up biochemically to identify the trigger for the conformational change. For example, for the protein you see down here, this is called pyruvate kinase, or CDC19. We found that the conformational change LiP detects is due to binding of a small molecule, this is called the fructose bisphosphate, to its allosteric binding site in cells grown in glucose. And this binding event triggers a conformational change, which then affects the active site and activates the enzyme. And what you see in green here, these are the peptides detected by the LiP readout as peptides that change upon the switch from glucose to ethanol. So what we saw here is that the LiP peptides were exactly located at the binding site of the allosteric regulator and the active site of the protein. And why I show this experiment, which is rather old, is because from this picture we had an idea. And the idea was that the method could be used potentially to detect protein small molecule interactions, since it detected the interaction you see here. At that time, we thought this could be a pretty simple experiment. We could lyse cells, any type of cells, yeast cells, bacterial cells, human cells, extract proteins under native conditions, and then add a compound to the cell extract. And then we could compare proteolytic patterns for as many proteins as we can detect in the presence and absence of the compound. And our idea was only those proteins that bind directly the compound should show an alteration in their LiP patterns, minimally at the binding site. In case of allosteric events, we could also see alterations at other regions. So that was the idea. And then we started to test our idea with a number of model compounds. And here I'm showing data for ATP. This is a very promiscuous molecule in our cells. It's known to bind many proteins. And the second compound is phenylalanine. It's an amino acid, and it's known to bind only 15 proteins in bacteria. So these are data from E. coli cells. And what you see up here is volcano plots that show you in the top left and top right boxes proteins that significantly change their proteolytic patterns upon addition of the compound to the extract. So for ATP, as you can see, we detected many proteins, among which several kinases and phosphatases and proteins that were known to bind ATP. For phenylalanine, way less proteins, among which 3 of the 15 known interactors of phenylalanine and a few new ones. So what happens if you look at a drug? I will now show you the example of cerulenin. This is an antifungal compound. It's known to bind covalently a single protein, fatty acid synthase subunit 2. So in this case, we added cerulenin to the extract of yeast cells, and we measured proteolytic patterns for 2,000 proteins. Out of these 2,000, one changed proteolytic pattern, and this was the known target of the protein. But what was peculiar about this experiment is that we looked at the peptides that changed, and we saw that a single peptide changed in the whole proteolytic pattern of the protein, and this peptide was precisely located at the known binding site of the drug. So next we asked whether this was a lucky shot or whether the technique could pinpoint binding sites of compounds. We looked into a few more cases. These are just some examples. And we saw that in the majority of cases, we could detect a change in a peptide located in the proximity of the binding site. Now, down here, you see the overall statistics. We looked at 40 cases of interactions that were known from the literature and for which a structure was available. In the majority of cases, the distance between the peptide we detected and the known binding site of the compound was below 4 angstrom, with some exceptions. So you see also the 4 guys up here, and we don't know exactly what these are. This could be false positives, or it could be also cases of allosteric interactions. Then what happens if you add increasing amounts of a compound to the extract? You see here the case of ATP, which we added at a concentration ranging from 1 millimolar to 25 millimolar. And what you see here in red and green are known ATP interactors. So as you can see, if you go from 1 to 25 millimolar, you recover more and more of the known interactors. And we learned later that you start to recover low affinity interactions. What happens then if you plot the intensity of peptides at the known binding site of the compound against the concentration range of the compound you add? Well, you see curves like the ones you see here that are very much reminiscent of binding curves. And the affinity values you derive from these curves are very close to the values that were available in the literature for the compounds we studied. So from these experiments, we learned that this approach enables the unbiased identification of protein small molecule interactions. At least in some cases, it pinpoints binding sites. And you can also derive quantitative parameters of binding. So having this approach at hand, we thought we should now try and think big. So can we come up with a map of protein small molecule interactions? We focused here on endogenous metabolites because identifying protein metabolite interactions is extremely difficult, and it's typically done via hypothesis-driven approaches where you have a hypothesis about an interaction between a protein and a metabolite, you reconstruct the interaction in vitro, and then you assess whether binding of the metabolite changes, for example, activity of the protein you're looking at, if it's an enzyme. Also, metabolite-protein interactions are very functionally relevant because they can alter enzyme activity dramatically. So we asked whether we could identify protein-metabolite interactions on a large scale. And we focused here on a set of 20 metabolites. These were intermediates from central carbon metabolism, amino acids, nucleoside phosphates, and also CAMP as a prototype for a signaling molecule. So it's basically the red compounds you see highlighted here on the right part of the slide. And then we added each of them to the extracts of E. coli cells. So this is again work done on bacteria. Some compounds were very promiscuous, like the one you see here. Some others seem to be very selective, like the one you see down here. And overall, we came up with a map of 1,600 roughly metabolite protein interactions, of which 1,400 were new and 230 were previously known. Then we looked into the known interactions, and we found that some of these were known regulatory interactions, for example, allosteric interactions occurring far away from an active site, and others were known catalytic interactions, so interactions occurring at a known active site between an enzyme and its substrate or product of the reaction catalyzed by that enzyme. So then we asked, can we use this information to classify novel interactions into these two types of groups? Well, we know the position of the binding site, so we can tell whether an interaction occurs at a known catalytic site or far away from it, at least for those proteins for which a structure is available. And just based on this very simple principle, we classified novel interactions as new putative catalytic interactions and putative allosteric interactions. And then we picked interactions from each of these two groups and reverted to in vitro assays to validate them. Now I'm showing here the example of GTP, which we found to be a putative new substrate of the enzyme phosphofructokinase subunit B. And we reconstructed the reaction in vitro and tried to hypothesize which products could be produced. And then we used untargeted metabolomics to show the disappearance of GTP and the appearance of the products we suspected. This is instead examples of how we validated regulatory interactions. And for that, we used classical activity-based assays. So what did we learn from this exercise? We learned that metabolite-binding proteins cover a broad range of functions. They are not only restricted to metabolism. However, within metabolism, they preferentially occur in the subnetwork involving the metabolite. For example, this is the case of PEP, phosphoenol pyruvate. This is the PEP biosynthetic step. And for PEP, we found mostly interactions upstream and downstream of the PEP biosynthesis step. We also looked into core proteins. Core proteins in E. coli are proteins that never change in abundance. The levels stay very constant, whatever you do to E. coli cells. The group of Bernard Paulson has looked into that, and they came up with a list of core proteins. And these proteins were enriched in metabolite-binding proteins, suggesting that transcriptionally stable proteins may not respond to perturbation by changing their abundance, but possibly by binding metabolites and therefore altering their activity. We also looked into enzyme promiscuity, so that means into those enzymes that, based on our data, bound many compounds. And we discovered that those interactions preferably occur at known catalytic sites, suggesting that enzyme promiscuity is largely due to substrate ambiguity, meaning that enzymes can possibly catalyze more reactions than we think. And this slowly brings me to a summary. So we learned that LiP-MS can detect protein conformational changes ranging from subtle to very large. It can also pinpoint the region involved in the conformational change. It can also identify structural alterations due to binding of small molecules and pinpoint, at least in some cases, the binding site. And it can also detect transitions between a protein and its aggregated state, like in the case of alpha-synuclein. What I did not show in this presentation is that it can also follow the unfolding of a protein directly in a cell extract. For example, you can lyse cells and apply a denaturing agent that could be temperature or guanidinium, urea, you name it. And then you can follow the decrease in intensity of fully tryptic peptides over the gradient of the denaturing agent you apply, and this results in unfolding curves for the proteins you can detect in the extract. And with this, I'm done, and I would like to thank people who generated the data I've shown today. So this was mostly work done by Johan Feng, Giorgia De Franceschi, Abdullah Karaman. They all left the lab and moved on now, and also Liliana Malinovska, Ilaria Piazza, and Valentina Cappelletti. These are our collaborators and our funding sources. And thank you very much for your attention.
Thank you very much, Paola. I want to hand over to Nigel Beaton. Nigel will show you how we developed within Biognosys the LiP technology to specifically be applied to the application of compound target deconvolution in mammalian cell systems.
All right, well, thank you very much, and thanks, Paula. That's super interesting, and so much hard work went into that. So yeah, I'll talk now about our work, our continuing development of LiP here at Biognosys. So we actually in-licensed the patent on the LiP technology from Paula's lab in ETH approximately 2 years ago, and at that time we had a couple of key milestones in how we wanted to continue to develop LiP. Our main focus was on establishing it for target deconvolution in mammalian systems. This is where a lot of drug development happens, and this to us presented a few key challenges. So one would be the increase in organismal complexity. So there's a larger proteome, and this also leads to a potential increase in noise and might make it a little more difficult to pick up the signal. So to address this, we've taken an approach that we believe allows us to achieve high selectivity, but coupling this with high sensitivity, and in this way we can identify candidates in an unbiased manner. So two of the key steps that we've implemented here at Biognosys are, one, we always do this approach now in a dose response style. So Paula touched on this for some of their experiments, and initially we were trying single concentrations, but we find that the dose response is a much more robust approach. This is probably not big news to anybody because I'm sure you've seen this before, but the principle is pretty straightforward in that if we're going to see a peptide that's generated by LiP, what we colloquially call a LiP peptide, there should be a proportional response in that peptide based upon the increasing concentration of a compound that is added to a lysate. And the second implementation that we did is we actually developed an in-house algorithm that was based upon extensive ground truth experiments. So these were always positive controls, well-characterized drugs where the targets had been confirmed by other methods. And we used these to actually establish what parameters drive a positive identification of a LiP peptide that is genuine. And ultimately, we trained these via machine learning, and we're able to select out the 4 key parameters that actually distinguish a known target peptide from either a false positive or perhaps noise. And so you can actually see those on the screen here. So the dose response is obviously a key driver. What you expect to see is that you get a genuine increase with compound in a LiP peptide. We also find that the differential Q value, so this is really just an FDR adjusted P value at the high concentration of the drug versus vehicle, plays a role in whether a peptide is identified or not as a positive hit. We've also put together what we call a PFL, which is a protein frequency library. And this is a library of common contaminants that we find in these LiP experiments. I'm sure many people are familiar with the crapome from immunoprecipitation assays. So this is along those lines. And we find that approximately 20% of proteins that we identify appear as a potential target in an experiment where we would not expect to see them, where the targets are known and they shouldn't be there. And this is to varying degrees. Sometimes they appear frequently. GAPDH would be an example of this, sometimes very rarely. And then the fourth parameter is multiple peptides. So in a genuine target, when you have a binding event, we would expect that it's more likely you're going to get more than one peptide from that protein than you would from just a random noise protein or peptide that shows up in your candidate list. So we actually aggregate all these together into what we call a LiP score, which we scale between 0 and 6. And you can see in the bottom now how these individual parameters contribute to the LiP score. So the dose response is the most powerful. It contributes about 2/3 of the weight to the LiP score, with the Q value, the protein frequency library, and the multiple peptides roughly contributing equally to the rest. So to test this, we've done pretty extensive testing, and I'll just highlight 2 cases that we've worked on today, and that's the kinases and the phosphatases. These were interesting ones for us, obviously, because they're key to life, all sorts of signaling pathways, but they're also really hot drug targets, in particular in cancer. And in the kinases, I'll show 2 compounds that we worked on, staurosporin and selumetinib, and we selected these because of their very different mechanisms of action. So selumetinib is highly specific. It really only binds 2 known kinases in the MAP kinase family, whereas staurosporin is broadly unspecific and binds tons of kinases. So this allows us to highlight 2 different sort of modes of action. And within the phosphatases, I'll focus on 2 compounds, colliculin A and phosphatase. And we selected these compounds because they target the same family, that's the protein phosphatase family, but they actually specifically target different members. So colliculin A is known to bind phosphatase 2 and phosphatase 1, whereas phosphatase does not target or bind phosphatase 1 at all, but binds 2, 4, and 6. So I'll focus on the kinases first. So selumetinib, as I mentioned, is a MAP kinase inhibitor. And across an experiment, we detect typically about 100,000 peptides. This is all in HeLa cell lysate, coming from 5,000 to 6,000 proteins. And if we run our algorithm, generate a LiP score across those 100,000 peptides, and then rank them based upon this LiP score, what we find is the top 15 from the entire experiment. We have 4 peptides from the MAP kinases, 2 from MAP kinase 1 and 2 from MAP kinase 2. So this is actually a really nice enrichment for the known targets within this candidate list. And you can see on the right-hand side of the screen what the dose responses for 2 of these look like, and they're pretty nice. So you see a genuine increase as one would expect. If we look at staurosporin, which I mentioned is acting by a different mechanism, it's a much more wide kinase inhibitor. What we find is that across the data set in the top 15, we actually pick up 13 different kinases in the top 15 peptides, and these map back, I believe, to 10 different proteins. So just confirming that it really is a broadly acting drug, and we're actually able to detect this unspecific nature with our assay. And again, you can see on the right just 2 of those selected peptides and how they behave with increasing concentrations of staurosporin. So those are just the top candidates. If we take a step back and look at the data set as a whole, what we find is that we really do strongly recapitulate the binding affinities or modes of action of these drugs. So you can see on the bottom left a jitter plot which shows every peptide that was mapped within these experiments, staurosporin on the left and selumetinib on the right. And we have a threshold of LiP score of approximately 1.5, above which we consider to be really potential genuine targets. I don't have time to go into how we came to that number, but that's based upon a lot of background. And if we look above this threshold for staurosporin, we find that 43% of the peptides that we map are from kinases, and they map to 21 different proteins. So this is a significant enrichment versus the proteome as a whole. Whereas on the other hand, if we look at selumetinib, in this target space, we actually find that the only kinases we pick up in red there are MAP kinase 1 and 2, so it's really highly specific. This is summarized in the donut plots on the right, where you can see that for selumetinib, we only pick up MAP kinase 1 and 2, whereas for staurosporin, although we do pick up the same kinases, we pick up a whole bunch of individual ones, but across the entire data set, we have an equal distribution of kinases in both experiments. So I think this shows the modality of these drugs really nicely. Something that probably was noticed by someone out there already is that in those data sets, there were some other proteins popping up, of course, and there was one in particular that caught our eye. It's a non-kinase target, NQO2. This is a quinone reductase, and it demonstrates the properties that we would expect from a genuine target, in that the peptides rank very highly. There's multiple peptides, and they show really strong dose-response correlations, but it's not a known binder of these drugs. But when you do go to the literature, what you find is that this has actually been shown to be a common off-target for multiple other kinase inhibitors. So what I'm showing here is for crenolanib, which is a receptor tyrosine kinase inhibitor that's been shown to bind NQO2. So we believe that this is one of the really nice attributes of LiP, and for these 2 compounds, the first case where we're able to show pretty strongly a new off-target for these drugs. So I'll move on now to the phosphatase data set and start with colliculin A. So for colliculin A, the targets are protein phosphatase 1 and protein phosphatase 2, their family members. And what we find in here is this is a really robust experiment, that the top 15 candidates across the entire target space are all from either members of this family. And you can see in the dose responses on the right that these ones are kind of like textbook dose responses. They look really, really nice. So we really robustly pick up the targets of colliculin A, which is nice. But if we move to phosphatase, which we selected because of its overlap and lack of overlap with colliculin, where we expect to pick up protein phosphatase 2 and 4, but not 1, this is actually exactly what we find in the data set. So we find that among the top 15, we really robustly identify protein phosphatase 2 as a target, also with peptides from protein phosphatase 4 and 6, but no peptides above our LiP threshold actually from protein phosphatase 1. So we completely miss this protein, which is what we would hope to find. A nice additional attribute of our LiP approach is that we're able to map a relative binding affinity. So we call these for now LiP IC50s. And what we find is within this data set, we actually recapitulate what is shown in the literature. So it's known that colliculin A has a higher affinity for protein phosphatase 2 than 1, and that's exactly what we find in the data set. And for phosphatase, that there's a similar affinity between protein phosphatase 2 and protein phosphatase 4, and this is recapitulated in our data sets. One thing I would note is that we find in our LiP approach that we consistently predict an IC50 value that's roughly an order of magnitude higher, which we think is just an intrinsic thing with the approach and all the steps necessary to generate a proper LiP peptide. But this is very consistent across all our data sets. And we really think the focus here is more on the relative affinities. And this is very useful for cases like this, where you're mapping out family members, or potentially when you're trying to factor in off-target proteins. Another really nice thing about this data set, I think, is it shows the consistency and the robustness of the approach. So colliculin A and Phosphatase are two completely separate drugs targeting some of the same and some different proteins. But what we find is in the same protein, so protein phosphatase 2 that they target, we very frequently generate the exact same peptide. These peptides rank equally high in the candidate list. And the thing I think is the most interesting is they show consistently the same directionality. So this to me shows that this approach is really robust. You have a binding event, you have a very specific interaction that occurs, which generates a novel cut site, and this happens across multiple peptides with multiple drugs. So this is really nice to see. And another thing we did, and this goes back, Paula showed a lot of really nice data about this, is across this data set and others, we've again found that taking this peptide-centric approach enables binding site confirmation. In this case, confirmation, in other cases, potentially prediction. And if we take the highest ranking peptides, what we find is that they very frequently map to the compound binding site. So on the screen now, you can see for 3 of the protein phosphatases with colliculin in the top left, and then Phosphatase, you can see the compound bound in blue for Phosphatase with the top ranking peptide in green, just flanking right beside the binding site. And I think this is probably the best demonstration of it is actually, if you look a little more at protein phosphatase 2, and you take the top 6 peptides based upon their LiP score, you find that these form basically a groove right through the protein, and that's exactly where colliculin A sits in the protein. So we're not just mapping a single one here, we're really recapitulating the entire binding site, which I think is really strong evidence of the value of peptide-centric approach. So I just wanted to show a few test cases, but just to wrap up, so now here at Biognosys with the Target Deconvolution, we've successfully performed this in a variety of organisms with many different compounds. So organism-wide, we've done several human cell lines, including primary cells, as well as yeast, bacteria, and fungi. And in a mammalian lysate with a cytosolic target, which is where we have the majority of our data, we have about a 2/3 success rate. So we're approaching 70% of the cases where we're actually able to pick out the known target from that complex lysate. And also want to bring it back to some of the advantages of the peptide-centric approach. IC50 estimation can be of great value when you're talking about families of proteins and trying to really figure out what might be off targets and what's not. But I think the strongest advantage here is binding site confirmation and prediction, which Paula has shown really nicely in her lab before. And then we've actually been able to extend and show again now in mammalian lysate. And on that note, I will say a couple quick thank yous. So to everyone at Biognosys, because basically everyone contributes to this project, but specifically Lucas and Roland, who have a heavy hand in this project. And then also Paula's lab, who have been great collaborators, especially Ilaria, a postdoc, who we work closely with.
Thank you very much, Nigel. And again, thank you, Paola, for these 2 presentations. And here at Biognosys, we are, as you can see, very excited about the technology, about what can be achieved today already internally and with our customer projects that we are running. And I think there's many more things to do in the future. Thank you. With that, we close the session, and I wish everybody a nice day or evening.
Thank you.
Thank you.